Good afternoon, everyone. First, thank Susan and Nico for organizing such an event to discuss sigma-2 receptors. My topic is the single nucleus mRNA analysis of sigma-1 receptor TMEM97 PGMC1 in postmortem brains. Sigma-1 receptor has a radioligand potassium, and the guinea pig brain is the source for sigma-1 receptor. Sigma-2 receptor, the traditional radioligand DTG, it has binding affinity for both sigma-1 and the sigma-2 receptor, but the sigma-1 receptor can be blocked using potassium. IgM1 is a novel radioligand developed by our group. IgM1 means RH-MAC. Sigma-1 has been cloned. Sigma-2 was cloned 20 years later after sigma-1 receptor being cloned. So our group has identified the PGMC1 complex as a putative sigma-2 receptor binding site. We use the IgM1 based affinity probe. And this PNS paper is using different ligand and use affinity column purification method. And they also identified the amino acids mutation can abolish DTG binding. So that's the two amino acids contributed for binding sites of sigma-2 receptor. So along of Andre Gross group, this paper is very important, very interesting, but there are still some questions. So is the TMEM97 gene encoding sigma-2 receptor in the human brains? So we will discuss this in two aspects, from protein expression and mRNA analysis. Sigma-2 receptor in cancer has been well reported. It's highly overexpressed in a variety of solid tumor cells and highly expressed in proliferation cells. And the sigma-2 receptor can be a good target and a good biomarker for cancer proliferation. But the sigma-2 receptor in human brain are not well studied. If you use the DTG autoradiograph, this is the first publication. It showed the DTG binding is quite lower, and DTG is not selective. So there are still questions about the results, how to interpret their results. And the ceramazine is another radioligand used for a human brain autoradiography and binding assay. So based on the binding assay, ceramazine has a high binding affinity, but the binding to the human brain homogenator capacity Bmax is moderate. Also determined that the DTG binding affinity were lower in human brain tissue. So this further confirmed the DTG is not ideal radioligand for human brain autoradiography. So IgM1 binding affinity is high in rat liver homogenates, also non-spec binding were lower. And when we compare the DTG binding profile and the IgM1 profile, we use a panel of compounds with different binding affinities. So if we put a binding affinity together, the affinity from DTG and the affinity measured from IgM1 actually are very consistent, and this unit line. So the advantage of IgM1 is it's a high affinity ligand for sigma-2 receptor, and it's selective for sigma-2 receptor. So it has a slow dissociation rate. So it can serve as a radioligand for autoradiography for human brain. So our group has extensive experience with autoradiography method, quantitative autoradiography method of different biomarkers. So this cartoon is very crowded with a lot of information from dopamine D1 receptor, D2 receptor, D3 receptor, and transport for dopamine and transport of monoamine. So we have our technology. So our question is, what's the densities of sigma-1, sigma-2 receptors in human brain? So first we conducted the saturation binding assay. We determined the potassium binding affinity to be about 4 nanomolar in human brain frontal cortex. And IHM1 also has high binding affinity, about 7 nanomolar in the frontal cortex of human brain. And we also use the potassium to measure the different brain regions to measure density of sigma-1 receptor in human brain. So top is the total binding using potassium, tritium labeled potassium. Bottom is the non-specific binding defined by blocking using 10 micromolar haloperidol. So that's the binding density of sigma-1 receptors. Sigma-2 receptor density in human brain, we measured using IHM1, and the non-specific binding was defined using 1 micromolar thyramycin. And that's all of the density summarized, the density measured from different brain regions, frontal cortex, striatum, and the globus pallidus, substantial nigroid nucleus. And it's very interesting. There's very good partial correlation between sigma-1 and sigma-2 receptors. So this is an example in the frontal cortex. So the correlation is the linear partial correlation and the R-square and the 0.68. So if we compare sigma-1 receptor and sigma-2 receptor, we found that sigma-2 receptor density is higher than sigma-1. For example, here, sigma-1 is about half of the sigma-2. That means sigma-2 are 2 times higher than sigma-1 receptors. So we are focused on striatum. So those are representative images from IHM1 binding and the potassium binding for sigma-2 and sigma-2 receptors. So what we found is that sigma-1, sigma-2 receptors extensively distributed in different brain regions. And next, we want to conduct the mRNA analysis of those receptors, sigma-1 receptors and sigma-2, because sigma-2 is identified as TMEM97. Also, we found PGMC1 also may contribute to the sigma-2 binding site. So that's why we conduct a systematic analysis of those mRNAs from those biomarkers. So we have a frozen brain, then we take out the putamen and we purify the single nuclei and prepare suspension. Then furthermore, we load the suspension to 10x genomics workstation to label the nuclei. And then we amplify the cDNA and construct the library for sequencing. After the sequencing, we do bioinformatics analysis and we can cluster different cell types. So we started 12 cases of putamen samples from 4 healthy control for Alzheimer's disease and for Parkinson's disease, 6 males, 6 females. They aged from 69 to 85 years old. And we collected more than 30,000 nuclei from those tissues. And of course, we have more to be analyzed for deep sequencing and for different brain regions and for different, even more cases. So this is the data for sigma-1 receptor nuclear mRNA. So that's classed, this map. So this information is easy to understand. So basically, sigma-1 receptor mRNA expressed highly in oligodendrocytes than neuron, but with high percentage of neurons express sigma-1 receptor mRNA. And astrocytes 1 is the resting astrocytes. Astrocytes 2 is the activated states of astrocytes. Neuron oligodendrocytes, astrocytes 1, also endothelial cell and parasites express sigma-1 receptor. TMEM97 nuclear mRNA has some similar to sigma-1 receptors. For example, it expressed in astrocytes 1, neuron oligodendrocytes. But the expression, both the percentage and the percent expression are all low in those cells. And TMEM even lower than sigma-1 receptors. But again, so sigma-2 receptor autoradiography, so the sigma-2 density is higher than sigma-1 receptor. So TMEM97 mRNA data is not consistent with autoradiograph data from sigma receptors. So the question is either TMEM97 the sigma-2 receptor in those brain tissue actually in putamen. We didn't have other data yet. So, but if we look at the PGMC1 nuclear mRNA, it's quite interesting. We see it's highly expressed in brain. And PGMC1 expressed in astrocyte 1, endothelial parasite, neuron and oligodendrocytes, and oligodendrocyte precursor. So there are more similarity with sigma-1 receptor. And PGMC1 also much higher, expression mRNA expression much higher than sigma-1 receptor. So we also analyze PGMC2 nuclear mRNA. It has a similarity with PGMC1 other than oligodendrocyte precursor. So I just summarize what we have done. So we identify IHM1 as a novel radioligand for autoradiography compared to the DTG. It's more with higher affinity, high selectivity. So it's much better radioligand than DTG for autoradiography start. And we also found that sigma-1, sigma-2 receptors are extensively distributed in different brain regions. Sigma-2 receptor density is higher than sigma-1 receptor density in most brain regions except the red nucleus. PGMC1 mRNA is higher than sigma-1 mRNA in human brain. So those are consistent with sigma-2 receptor autoradiography. TMEM97 mRNA is lower sigma-1 mRNA in human putamen. So yeah, we have more questions to ask. What's the binding sites of IHM1 in human brain? So is the TMEM97 a sigma-2 receptor in the human brain? So what's the relationship between PGMC1 and the sigma-2 receptor? So what's the functions of those receptors in Alzheimer's disease or Parkinson's disease brains? So I would like to thank my collaborators and the funding source and the Genome Sequencing Center, Genome Science Center, ADRC, MDC, and all the participants and the donors for donate their brains for our study. If you have more questions, want to discuss, my email address is here. Thank you very much.